Good evening, dear students. Today we will going to discuss about the basics of the plant tissue culture. Before explaining the all the details of the plant tissue culture, we will discuss about the basic definition of the plant tissue culture and the history of the plant tissue culture. So plant tissue culture is an in vitro culture or growth of the cells, tissues or the organs of the plants in a sterile condition and well formulated media to produce an entire plant. In vitro stands for the in glass, that is the culture of the cells or the any part of the plant in the test tubes. Now, history of the plant tissue culture. German botanist G. Haberlandet is regarded as the father of the plant tissue culture. In 1902, he was regarded as the father of plant tissue culture. He has done his work from 1845 to 1945. Now, basic steps involved in the plant tissue culture. Here is a basic protocol of the steps which are involved in the plant tissue culture: preparation of suitable nutrient media, selection of explant, sterilization of the explant, incubation, transfer of the callus and regeneration, hardening or acclimatization, in the end transfer of the plantlets into the field. Now, preparation of the culture medium. There are so many culture mediums available in the market which are used in the plant tissue culture. MS medium, White's medium and B5 mediums. These are very popular nutrient medium which are used in the plant tissue culture method. These contains inorganic nutrients as well as the organic nutrients. Two types of inorganic nutrients are used in the culture medium: 6 macronutrients and 6 micronutrients. 6 macronutrients are like nitrogen, phosphorus, sulfur, calcium, potassium and magnesium. 6 micronutrients like iron, molybdenum, copper, zinc, boron and manganese are also added in the medium. Organic nutrients, carbon and nitrogen containing compounds are used. Sucrose and fructose and other carbohydrates are the carbon source. Now, selection of the explant. Explants may be any part of the plant, that are root tip, shoot tip, anther, ovule, ovary. As you can see the picture, there are some root tips and shoot tips can be seen. Sterilization of the explant. Sterilization is very important for the plant tissue culture. Not even explant, we have to sterilize our equipment which we will use for the plant tissue culture. There are so many chemicals available in the market such as sodium hypochlorite, silver nitrate, calcium hypochlorite, bromine water, mercury chloride, hydrogen peroxide. And in the colleges and the schools, laboratories having ethyl alcohol, which is the very most common sterilizing medium which is used for the plant tissue culture. Now, inoculation. Inoculation means the sterilized explant is now added into the nutrient medium. That is the inoculation. The inoculation is done into the aseptic conditions. That means there is no presence of the microorganisms. So, as you can in the picture, there is a laminar airflow cabinet is available for the method of the inoculation. Basically, laminar airflow having filtered air which helps in the prevention of the growth of the microorganisms. Now, incubation. Incubation is done at 25 to 28 degrees Celsius with a relative humidity 50 to 60% with 16 hours of the photo period. After the period of the incubation, an unorganized and undifferentiated mass of the cells called callus is obtained from the each explant. In third picture, we have seen the callus, which is an undifferentiated mass. In some cases, roots and shoots directly develop from the explant after the incubation. Now, transfer of the callus and regeneration, or this is also called as the subculturing. The callus, which is obtained from the incubation of explant, is transferred into another medium and reincubate. The callus, firstly the callus which grows, that takes up all the nutrients, then we have to transfer into new nutrient medium so that callus can be differentiated into roots and shoot and can be easily grown into the new plant. So basically it is called as the subculturing. That means the transfer of the callus into the new nutrient medium. Now plantlet regenerates from the callus with the introduction of the roots and shoot directly from the callus. Hardening or acclimatization. In picture D, there is a hardening and acclimatization occur so that the plantlets can be adjusted according to the environmental condition. Now these small plantlets start grow, then these are going to transfer into the plantlets to the field. After the hardening process, plantlets are transferred to the either greenhouses or the field conditions outside. These plantlets will grow into independently and healthy plants. Now, importance of the plant tissue culture. A large number of plants having identical features to the parents can be produced by this method. Adult plants can be produced with the short period of time. Many plantlets can be produced without seeds. Healthy and disease-free plants can be propagated by this technique. Endangered plants can be preserved or conserved by this technique. Thank you.